Dysfunctional autophagy stands at the crossroads of many age-related diseases, emerging as a critical factor that links cellular decline to the chronic conditions that burden so many older adults. When autophagy falters, whether through genetic predisposition, lifestyle factors, or simply the wear of advancing age, cells lose their ability to efficiently remove damaged proteins, malfunctioning organelles, and toxic aggregates. This buildup of cellular waste doesn't stay isolated in one tissue. It reverberates throughout the body, undermining systems and laying the groundwork for disease. In neurodegenerative disorders like Parkinson's disease, impaired autophagy leads to the accumulation of misfolded proteins such as alpha-synuclein, which aggregate into Lewy bodies and disrupt neuronal communication. Healthy autophagy would normally recognize and clear these aggregates before they can damage brain circuits responsible for movement, balance, and coordination. Instead, when autophagy becomes sluggish with age, these toxic proteins accumulate unchecked, accelerating the death of dopamine-producing neurons and driving disease progression. Enhancing autophagy through experimental compounds or lifestyle interventions shows promise in animal models for slowing this process, raising hopes for future therapeutic strategies. In metabolic diseases like type 2 diabetes, dysfunctional autophagy disrupts the delicate balance of insulin production and sensitivity. Pancreatic beta cells, which produce insulin, rely on autophagy to maintain mitochondrial health and manage endoplasmic reticulum stress. Without effective autophagy, these cells become overwhelmed, leading to impaired insulin secretion. Meanwhile, in tissues like liver and muscle, reduced autophagy contributes to insulin resistance by allowing lipid droplets and dysfunctional mitochondria to accumulate, disrupting glucose uptake and metabolism. Together, these changes drive the hallmark high blood sugar of diabetes, but studies show that restoring autophagy can improve insulin sensitivity and beta cell function. Cardiovascular disease also has deep ties to impaired autophagy. Healthy autophagy in heart muscle cells, cardiomyocytes, helps clear damaged mitochondria and protein aggregates that could compromise contractile strength and electrical stability. When autophagy is insufficient, these damaged components accumulate, increasing oxidative stress and inflammation in heart tissues, key factors in the development of heart failure and arrhythmias. Similarly, impaired autophagy in vascular endothelial cells leads to chronic inflammation and plaque formation in arteries, accelerating atherosclerosis. Enhancing autophagy in cardiovascular tissues has been shown in preclinical studies to reduce plaque buildup, improve cardiac function, and limit heart muscle damage after stress events like heart attacks. What makes these connections even more compelling is that interventions boosting autophagy can target multiple diseases simultaneously. Lifestyle changes like intermittent fasting, caloric restriction, and exercise not only slow age-related decline, but also show broad protective effects across the brain, pancreas, heart, and blood vessels, precisely because they restore or enhance autophagy in these tissues. Certain experimental compounds, such as spermidine and rapamycin analogs, are being actively studied for their potential to pharmacologically activate autophagy and mitigate these diseases. Altogether, the realization that impaired autophagy is a common denominator among many of aging's most devastating diseases highlights its importance as a therapeutic target. Rather than approaching each condition in isolation, supporting autophagy offers a unified strategy to preserve cellular health, slow disease progression, and extend not just lifespan but health span, allowing more people to age with vitality, independence, and quality of life.